It is a real pleasure, and I'm excited to talk about extreme environments on Earth. And first, as we've heard, there is several talks about those. So I give you a little overview, a brief overview, before I then concentrate on just one extreme condition, and that is, in my case, low pressure, because after as soon as I came to Florida, I basically started working with hypopiezotolerant microorganisms. And it is a very small area, and I would like to introduce to you those kind of bacteria and suggest them as potential model organism in astrobiology. Okay, so it's not moving. Oh, here we go. So first, we need to define what is an extreme environment. And there is one definition that basically says an extreme environment is an environment that exhibits conditions which are challenging for most life forms. This is a very anthropological point of view, because we as humans, we kind of are adapted or are able to live in mostly moderate conditions. And when we basically look at extreme conditions, then for us, it is just slightly different, versus when we go to, for example, very high temperatures, there is life forms that can grow in these extreme temperature. They actually would not be able to survive or have a hard time growing at moderate temperature conditions. So it's always a different point of view. So we just continue with our point of view, what we consider extreme. First, I would like to take you on a tour through Earth, and Earth is amazing. We do find a lot of different extreme environments, and one of those extreme environments is seen in Iceland. What you do see here are hot sulfur springs. The name already says it. We reach temperatures up to boiling, 100 degrees Celsius. We see a lot of metallic compounds, like a lot of sulfur, which are pretty much toxic for a lot of life forms, and the environment is very acidic. So any life form that is adapted to those kind of conditions have to cope with a lot of different stressors. Now, when we look at the other extreme of the temperature scale, we kind of reach cold temperatures. This could be either like in the high alpine regions, where we have a snow environment where the snow cold is more temporarily, versus when we go look even further towards glaciers and permafrost, where we have basically an icy environment throughout the year. These environments are characterized by low water availability, which is strange, because there is actually quite a lot of water around. It is just in ice form, and hence it is not available for the different life forms to actually use and grow in these environments. They're also characterized by low nutrient availability, and at high altitudes, like we've seen here, where we have a snow cover, bacteria or life forms also have to deal with freeze-thaw cycles throughout their lifespan. So now we've been at the peak of the Earth, let's go down to the bottom. What you see here is a black smoker at the Mariana Trench, at the bottom floor of the ocean, which is one of the deepest parts of the world. We have very high pressures. We're talking about 120 megapascal. So the bacteria or life forms have to deal with these high pressures. Not only high pressures, but you see the fumaroles coming out of those black smokers. They contain high sulfates. They're very acidic, and they're actually very hot. They can also reach up to 100 degrees. So any microorganism that is kind of adapted to those conditions has to not only deal with these conditions, but there's also no sunlight, and there is mostly an anaerobic environment, which means there is barely any oxygen available. And you would think, oh my God, that is really, really extreme. But as the picture already displays, we do see some life forms growing on those black smokers. So they apparently are perfectly adapted to these kind of extreme, what we consider extreme environment. So I've already talked about some of those environments that they were quite acidic. When we look at the other end of the pH scale, we have a very alkaline environment, and we do find those naturally occurring on Earth as well. So one example is Mono Lake in California, and Mono Lake contains a lot of carbonates, which leads to a pH of about 10, like regular water is pH 7, the scale ends at 14. We also know that Mono Lake is about 3 times as salty as the ocean. So again, a lot of low water availability and high osmotic pressure. So this is just like things life forms have to cope with. And when we can even go more extreme when it comes to osmotic pressure and low water availability. And these are what you see here is a salt mine down in Ulby in England. And this was one sampling event during the MACE program that I used to work in Edinburgh. You see on the right top corner, you see basically a man-made tunnel, and you see the salt deposits around here. And then at the bottom, you see the salt crystals formed between the interface of the brines and the surrounding environment. So we went there because we kind of wanted to know whether bacteria can grow in these extreme environments. So we went there, sampled the brines. Those brines are mostly anaerobic, and we took them to the lab and basically tried to find some life forms. Again, those environments are characterized by high osmotic pressure and low waterability. Low waterability, we also find those on in deserts. This is a picture I took at the United Arab Emirates. And deserts are usually known to be very dry. Also, bacteria or life forms that live there kind of have to deal with large temperature ranges throughout the daily cycle. We have about 40 degrees during the day, and at night the temperature can go down to about -4 degrees. So this is a lot to cope with for most of the bacteria. Also, it is known that there is not a lot of nutrient availability. So just in general, it looks like a very hostile place to be in. These are just like a few places. There's way, way, way more out there. But I just wanted to cover like one of the most important stresses and the most prevalent stresses here on Earth. Also, what we've seen whenever I talked about one single environment, they were mostly characterized by not only just one extreme condition, but by several. For example, the black smokers, we saw high temperature, high pressure, and high acidity. For Mono Lake, it was highly alkaline, and we've seen a higher salt concentration. So just to keep in mind, like when bacteria or when life forms grow in extreme environments, they usually have to be adapted to several challenges that occur there. So why are we actually interested in those environments? So when we go there and study those environments, we get information about the limits of the biosphere and the limits of life. So when we go there and we do not find life, that basically tells us, okay, we found like a barrier, a boundary. Beyond that, where there might not be life. However, if we do find life, that's positive too, because we can take those bacteria or life forms, take them to the lab, do some experiments with them, and find out why they are adapted, why can they grow in these extreme environments. And that gives us fundamental insights into the biochemical architecture of life. And once we found those specific features and once we found those adaptations, we can actually see if there are useful products that we then can use for Earth-based applications. And there is a huge list. This is just like one tiny table from industrial applications of archaeal products. There's way more out there. I want to just focus on one example. And these are like enzymes from thermophilic bacteria or archaea, and they have been isolated. They work better at high temperatures. They don't denaturate, as easily, and therefore they have been added to laundry detergents and basically improve their performance detergent. So this is just one way of how to use information and or these adaptations from life in extreme environments. But then we can also look further beyond Earth. Like we are interested in whether there is life outside Earth, is there life on extraterrestrial planets? And these extreme environments can serve as terrestrial analogs to study the limits of life elsewhere. And one of those examples is the Rio Tinto river in Spain. And it has been described as a geochemical and mineralogical terrestrial analog of Mars, because what we found there are like the chemical composition is quite similar and mimics Mars in quite a lot of ways. Obviously, it is not perfect. There is just no perfect Mars analog on Earth because there's certain environmental conditions on Earth that are not available on Mars, that are not available on Earth, but we can get as close as possible to those terrestrial analogs, and Rio Tinto with the color from the iron is very close to Mars. It's close to Mars. And, so by going to these extreme environments, we are basically able to define the boundaries of habitability and thus the prospects for life elsewhere. So then we need to look at who actually lives in those extreme environments. And we have found quite a lot of record holders on Earth. So here is the phylogenetic tree. We have the 3 domains, bacteria, archaea, that are grouped within the prokaryotes and are single cell organisms. And then we have the multi-cell organisms, which are called the eukaryotes. And when we look at these at the phylogenetic tree, we can find those extremophiles everywhere. Extremophiles are basically, the definition is they are optimally adapted and they optimally grow at these extreme environments. And they are found everywhere, but still archaea contain the most record holders. I do want to briefly point out one of my favorite eukaryotes, which is a tardigrade. You probably heard also its name is water bear or a moss piglet, and it's a very, very hardy animal. It has been sent to space. It has been exposed to the internet outside the International Space Station to space conditions. It survived. It can also survive about 10 years in a desiccated and dried state, and it can survive high doses of radiation. So this little animal is quite a very hardy microorganism and a potential model organism for astrobiology as well. And then here is just a table of all the different parameters of a lot of different stresses and just some examples of record holders for each of the different stressors. It's not a complete list, and I won't go into detail because there is more talks coming, which probably are going to cover most of those. As I said earlier, I am mostly focused on pressure, and that's what we look at today. So we do need to do some definitions first. So historically, life forms that were able to grow above 10 megapascal were called barophiles. In 1995, Janos proposed the word piezophile, from Greek piezo to press and philo to love. So we stuck to this word from now on, and they are defined, piezophiles are defined to optimally grow between 10 and 60 megapascals. Hypopiezophiles are grow optimally between 60 and 120 megapascals, and hypopiezophiles are at the other end of the spectrum, and they grow optimally between below 25 hectopascal. So then I would like to introduce the word tolerant. Tolerant bacteria are described as bacteria that have the ability to survive and may have the potential to grow in a certain defined range. However, they only survive. Like they don't necessarily, it's not their optimum conditions. So whenever they are outside their optimum conditions, but they still can to survive or grow, they are called tolerant. So when we look at the pressure and the record holders, we have hypopiezophiles. One of the record holder is Pyrococcus yayanosi, which can grow at 150 megapascals. When we look at hypopiezophiles, unfortunately, there is currently no known strain out there. And we do have an assumption why that is. And therefore, we need to look at the atmospheric pressures on Earth. The average atmospheric pressure is 1,013 hectopascal. When we look at the bottom of the Mariana Trench, where I've already shown you some pictures, is we have a pressure of 120 megapascal. On Mount Everest, the highest peak on Earth, we have 330 hectopascal. And then once we leave the atmosphere and we go into the stratosphere, we have about 10 hectopascal of atmospheric pressure, and above 80 kilometers, the pressure is reduced to below 1 pascal. And then when we look at interplanetary space, we reach almost vacuum conditions of a 1.32 times 10 to 7 minus 10 to 7 hectopascal. And basically what we've seen here is when we look at the hypopiezotolerant range, there is no such low pressure on Earth. Like these low pressure environments just don't naturally occur on Earth. And that might be also the reason why there is no bacterium that is perfectly adapted or like can grow under these conditions optimally, because they don't experience those conditions on Earth. So, but we are interested in not only boundaries and limits of life on Earth, but we also would like to see whether there is life somewhere else. And it has been shown that the Mars environment is the most Earth-like than that of any other planet in our solar system. It is still a very harsh environment there. It is not as habitable as planet Earth. We've seen most of the stresses there that I mentioned earlier. We also have a high UV radiation or radiation environment on the surface, and that is due to a lack of atmosphere. And because there is no atmosphere, we have only an atmospheric pressure of 7 hectopascal versus the 1,013 hectopascal we find on Earth. So that's why we are basically interested in whether bacteria can grow at low pressure, because if we go to Mars, if we are interested in whether life can exist, Earth-based life can exist on Mars, these bacteria need to be able to grow under low pressure. And with low pressure, there comes certain challenges. So we assume there is an impaired cellular structure and activities. We know that there is a set of selective stresses which may introduce so far unknown alterations. We also have only little data about on how the microbial metabolism is affected. And there's a lot of open questions because it is a very new area. There's not a lot of groups working on those bacteria. So there is open questions, but that leaves a lot of room for more research. And some of those open questions is, we actually don't know how low pressure is sensed in bacteria. We don't know how they acclimatize to pressure alterations. Maybe they do possess pressure-specific adaptations, which would be good because then we can look for marker genes. Are there marker genes? And that would help us to screen for hypopiezotolerant bacteria, or maybe there is metabolic pathways. There's changes in metabolic pathways. Maybe there is changes in the lipid composition. So far, there is just many open questions, and we have to start looking. Where do we look? Where we do know a little bit more about living under high pressure. So we just look what happens under high pressure, and maybe we can make some assumptions for what is going on in the low pressure. So the challenges are the same. We have impaired cellular structures and activities, and those are, for example, there is a tight packing of molecules, and therefore we have a loss of fluidity in the cell membrane. So which makes sense because the cell membrane is the interface between the high pressure environment and the inside of the cell. Like it makes sense that this is affected. We've studies have done where we looked at changes in gene expression, and we've noticed some changes in gene expression. There is also structural changes of biomolecules of the membrane structure. We've seen an increase in unsaturated fatty acids, which then leads or counteracts the loss of fluidity. We, in literature, the synthesis of pressure-inducible proteins have been described. They might be able to serve like as biomarkers, but in general, it is still comparatively poorly understood compared to other pressures or other stresses like high temperature or radiation. So now let's go back to our low pressure bacteria. So unfortunately, there is no true hypopiezophile known. But that is actually not too bad because we do have some hypopiezotolerant bacteria that have been described that are available for us in the lab that we can use as test organisms to find out what is going on in low pressure. So a total of 62 bacterial strains have been described by Andrew Sugar and Wayne Nicholson that can grow at Martian atmospheric pressure. And here is the microbial phylogeny and the diversity. These 62 are spanned within 3 phyla, the Firmicutes, Proteobacteria, and Archaetinobacteria, and they encompass about 10 different genera. Carnobacterium here and Serratia over here were the first hypopiezotolerant bacteria that have been described. So now once they've found those bacteria, they were wondering whether this is the genus-specific adaptation or whether it's species-specific. Because if it would be genus-specific, that would be perfect because then we can easily look at or can easily find more hypopiezotolerant bacteria. So then what they did is they studied 9 different type strains of Carnobacterium and 8 different strains for Serratia and exposed them to low PTA condition, low pressure. And what happened is in Carnobacterium, all of them were able to grow. Unfortunately for Serratia, only 6 out of 8 were able to grow at low pressure. So that means this adaptation is not genus specific, it is species specific. So, and that was basically the state of knowledge when I arrived in Florida. And my first task was to maybe we could find a few more. 62 is good, but maybe we could find a few more of those low pressure bacterium. And one place to look at is at clean room assembly facilities, where they basically put the Mars rovers together. Because if we send those rovers to Mars, there is quite a likelihood that there remains some bacteria on the spacecraft. This is also done in the, this is part of the planetary protection program. Like we need to make sure that we do not contaminate Mars with or other extraterrestrial planets with Earth microorganisms, which is called forward contamination. It also deals with backward contamination, which is just the opposite. If we take a sample from Mars, for example, and bring it back to Earth, we need to make sure that we don't contaminate Earth with any potential life that we bring back from Mars. However, like we try to be as sterile as possible, but it is just not, we are just not able to be completely sterile. So there's always a few bacteria there, but within the planetary protection guidelines, we go in there and sample those parts so that we get an idea if we are within the limits of the allowed bioburden within on a spacecraft and within a spacecraft clean room. And so to date, bacteria from actual Mars spacecraft and the surrounding clean rooms have not been tested for growth at low Martian atmospheric pressure. And so we got some bacteria from the InSight, the Phoenix, and a few other Mars missions. And we determined whether those spacecraft bacteria can grow at low PTA conditions. Low PTA conditions in this case are defined at 7 hectopascal, 0 degree, and an anaerobic atmosphere. Why is that? Like, the problem is at 7 hectopascal, water is not stable at room temperature. So when you look at the phase diagram of water, like the stability increases when you lower the temperature. And that's why we incubate at 0 degree, because we get a better stability of water. And since we already mimic Mars anyways, we decided, oh, let's just change the atmosphere to an anaerobic atmosphere and use CO2 instead of oxygen. So in the future, when I say low PTA, so these are the conditions: low pressure, temperature, and an anaerobic atmosphere. And then we also wanted to evaluate the effect on growth at low PTA conditions of a range of Mars-relevant geochemical electron acceptors. Like, we kind of knew it is going to be difficult to find new hypopiezotolerant bacteria. So we thought, like, oh, if we add a few other options for potential growth, maybe we get lucky and we will find one. So how do we actually grow bacteria at low pressure? Because I said we don't really have a naturally occurring low pressure environment on Earth. So in our lab, we had two different options. We have like one a Mars chamber, which is quite an elaborate piece of technology where we can simulate low pressure, but it also is a little bit more costly. So when we do high throughput experiments, we just use those desiccators that were modified. We basically attached a pump to those desiccators, which allow us to continuously reduce the pressure to 7 millibar and maintain it at 7 millibar, because water evaporation would increase the atmospheric pressure. So we were still able to pump this pressure out and maintain it at 7 millibar. We also have the option to flush it with CO2. We add some CO2 pouches so that we maintain an anaerobic atmosphere, and we have an indicator tablet in there that basically indicates via color change whether we have an oxygen influx or not. And we put all of those desiccators in a fridge and maintain the temperature at 0 degree. When we use those the regular agar plates, the only thing which we change is we pour them double thick, because as I mentioned earlier, we have some evaporation going on, and of the incubation, because they don't dry out as quickly. So we can actually put those plates in there for about 4 weeks. What you see here on the right is agar plate grown at optimal growth conditions. You see here actually the 8 different serratia strains that have been tested. This is optimal growth conditions. They all grow perfectly fine. On the right-hand side, you see bacteria that have the same bacteria that have been exposed for about 4 weeks at low temperature, low PTA conditions. And as I mentioned earlier, only 6 out of the 8 were able to grow. We also had some other controls on this plate. So that's basically how we are able to screen for growth at low PTA conditions. And what we've done with our spacecraft bacteria is I was able to get 125 different strains. We subjected them to the 4 different conditions to basically monitor what is going on when we add more and more pressures. We have the low PTA conditions, and then we have 2 stresses, 0 degree and CO2, and then we reduce it to just 1 pressure where we only have the low temperature, and control 3 is our positive control, optimal growth conditions. Here is a list at the bottom, is a list of the microbial diversity of the 125 spacecraft strains. Like, they span in this time 6 different phyla, and we have about 27 different genera that we tested. What you see here, the genera that are marked with the star are basically genera where we already have a known hypopiezotolerant bacteria. There has been hypopiezotolerant bacteria described in those genera, in this genuses. So in this case, maybe we get lucky and we find another one in one of those genera. And so what you see here is a subset of the result. I usually was able to test about 25 different strains per plate. We always did it in triplicate. You see here from left to the right is the same order of the controls, low PTA conditions here, reducing the amount of stresses and ending up with optimal growth conditions on the right-hand side. Each row basically are different nutrient conditions. We have the regular TSA plates here. We have TSA plates plus vitamins. We have TSA plus the nitrates, TSA plus sulfates, and TSA plus iron. So when we look at the positive control, we see that the addition of iron really reduced the ability to grow. We did not exactly expect that kind of result, but apparently iron has a negative effect on the growth of bacteria in general, even under optimal growth conditions. Then when we look from the right to the left, we realize that the amount of colonies per plate kind of decreases the more we add the pressures. That means we have distinct effects of pressure, temperature, and atmospheric conditions, and they are additive. We also seen that there is basically no growth at low PTA conditions, but when we took those plates, the growth resumed at optimal growth conditions. Basically, this plate came out similar to what we've seen at optimal growth conditions. What you see here too is we always need to add a positive control and a negative control. The positive control was serratia liquefaciens, and the negative control was a bacillus strain. And in all of those conditions, the positive control was able to grow. So summarizing, so we did not see any growth at low PTA, even so we had stable hydrated conditions, which are unlikely to even be found on present-day Mars. The bulk atmosphere on Mars plays a significant role in whether growth can occur, and the addition of different electron acceptors failed to stimulate growth. However, as I said earlier, we do know that there's something going on in those bacteria, because we do see that they can resume growth at optimal growth conditions after they were exposed to low PTA condition, which means that those low PTA conditions are not lethal for bacteria. What exactly is going on, if they're in a resting state or the metabolic activity is just so slow that we don't see growth within 4 weeks, we don't know. But there is inherent metabolic, genomic, transcriptomic, and proteomic constraints, but these are reversible. What does that mean for our journey to Mars? Well, in our case, from the ones we tested, these spacecraft bacteria are not likely to pose a serious risk for the forward contamination of Mars. So at one point, for habitability of Mars, the results were a little bit disappointing, and I did not get additional strains that can grow at low PTA, but for a planetary protection point of view, the results were pretty positive. So, but that wasn't a dead end, because we still have those hypopiezotolerant bacteria that have been described. And our favorite and model organism is serratia liquefaciens, because out of those, we know it can grow very reliable. It grows the best. It is a proteobacteria, it's gram-negative, and it is a rod, as you can see here. So I used serratia liquefaciens to basically get more information about what is going on within the cell and what is going on with their metabolism. And one way to study changes in metabolism is to characterize the carbon source utilization at low pressure. And we wanted to identify the effects of low pressure on metabolism, and we wanted to find unique organic substrates that are preferred by serratia liquefaciens for metabolic activity under those simulated Mars surface conditions. So how did we do that? we purchased Biology in 2 plates, which you see here at the bottom. It is a 96-word plate, and each of the 95 words contain unique carbon sources. I can group them in different substrate groups, and we tested basically about 28 different carbohydrates, 24 carboxylic acids, 20 amino acids, and we basically first did 2 experiments. The first one was we looked at pressure-only effects between 25 and 130 hectopascal. We could do that at optimal growth conditions because at 25 hectopascal, water is still stable. And then in a second round of experiments, we looked at the effects of Mars versus Earth pressure and changed the gas composition as well as the temperature. And then we're able to basically look at the different metabolic fingerprints at those different conditions. So how do we actually know that bacteria, that our Serratia used the different carbon sources? Each well also contains a redox dye, which is a tetrasodium salt. And once there is a reaction going on within the well, it is basically changing color from originally colorless to violet or purple, as you can see here at the bottom. And so we did that for our first experiments where we had 30-degree and oxygen environment, and we only changed the pressure. We did different steps, 25 hectopascal, 50 hectopascal, 100 hectopascal, and 200 hectopascal, and Earth normal pressure. And we, after the results came back, we did a principal component analysis, and the plot is basically what you see here. We see that the samples from the 25 and the 50 hectopascal kind of group together versus and are separated from the samples that were enriched at incubated at 100 and 200 and 1013 hectopascal. And basically what that tells us is there is something going on in the cell between 50 and 100 hectopascal. At 50 and everything that is to the left, we have a lower partial pressure of oxygen, and at higher, on the right-hand side, we have a higher partial pressure of oxygen. So there is definitely something going on where there is an inherent change in the metabolic activity. So actually, we can actually look at numbers. We were able, as I said, we can detect the usage of a carbon source by change of color, and we can measure the total well color development. And that's what we also did. At 100, at Earth normal pressure, we had a total well color development of 95 versus it has been reduced to by 75% to about 25 at 25 hectopascal. So that basically indicates a lower number, indicates we have less metabolic activity going on. The metabolism is slowed down. So when we look at the total amount of carbon sources used, Serratia liquefations out of the 95 different carbon sources that were tested, Serratia liquefations can use a total of 74 at optimal growth conditions, and the amount of carbon sources that they can use at 25 hectopascal is reduced to 41. So there is a big change going on, and we were interested, maybe we can find out what is the reason for this. So we looked at the different substrates group, and when we look at the carbohydrates, then we can see, for example, that there is barely any change. They were at optimal growth conditions, they were able to use a lot of carbohydrates, and that has barely changed when we look at 25 hectopascal. If we look at carboxylic acids, we see quite a reduction. They somehow lost the ability to use carboxylic acids at low pressure, and this effect is even more prevalent for amino acids. Originally, we had about 20 amino acids in the assay that were able to use around 16 at optimal growth conditions, and that number is reduced to 4, which is very unexpected. We don't really know why. We have some assumptions, but we don't really know why this is the case. It was very surprising. And then when we look at the low PTA conditions, we've seen a similar effect. The numbers are, what you see here, are even lower. If the trend continued, what we've seen, this line basically means that's when we put, after 49 days of incubation at low PTA, we put them for 2 days at optimal growth conditions to just see if they fully recover. They almost did. Instead of 47 down here at optimal growth conditions, they can use 64, even though they were exposed to low PTA conditions for 49 days prior to that. So that basically means most of these metabolic pathways are not permanently damaged and they can recover. So the take-home message from this experiment was that serratia liquefaciens has a high versatility at optimum growth conditions when it comes to usage of different carbon sources. At low PTA conditions, we saw a cumulative impact of the atmospheric pressure, the temperature, and the atmospheric composition. What we see from literature, when we compare that to what is available in literature, we saw that the ability to use carbohydrates and the inability to metabolize most amino acids under low PTA conditions is consistent with transcriptomics data. There has been done transcriptomics at, I think it was a Carboxylic, and they found an upregulation of genes in carbohydrate metabolic pathways and a downregulation of genes in amino acid pathways. So that is basically consistent, and there is so much more we need to look at. But I want to make the point that hypopiezotolerant bacteria should be potential model organisms for astrobiology, mostly when we look for potential life on Mars. So here is just a list of articles that came out that we published over the last 4 years. I want to point out this one. This is a review article because I was not able to cover everything what we know. So if you are interested, just it's free. You can have a look and get the full overview of what is currently known about hypopiezotolerant bacteria. I would like to acknowledge Dr. Andrew Sugar from the University of Florida, who was my boss. I would like to thank my two students, Mary Elizabeth Jobson and Rachel Tucker. I also want to thank Professor Wayne Nicholson from the UF for providing us with some strains, Professor Charles Coquel and the MACE team that basically supported me prior to this research. Also, I would like to thank the Planetary Protection Research Program for funding this research, and Florida Space Grant Consortium for supporting the students. There's one extreme environment I have not mentioned yet, and that is the International Space Station. And what we see here are 4 extremophile humans. And today there is a launch at 6 p.m. Central European summertime, where they launch on board a Falcon 9 in a Dragon capsule to the International Space Station. And where they are basically going to experience some extreme conditions. Stay tuned. You can find a live stream usually on the SpaceX Twitter page if you're interested and want to follow those 4 astronauts to launch today from Cape Canaveral here in Florida. So I'm super excited. I'm going to go and watch that launch after our talk in about 2 hours. And thank you very much for listening. Let me know if you have some questions. I'm happy to answer them.